Hi everyone, my name is Lindsay and I will be presenting my work investigating DNA damage responsive long non-coding RNA, supervised by Dr. Tharmalingam. This project was funded by NHRC and the session has not received any financial or in-kind support. The central dogma theory is one of the most critical theories of molecular biology. It describes the process in which DNA is transcribed into a messenger RNA, which is then translated into a protein. However, it only accounts for a fraction of what happens when DNA is transcribed. Of the 70 to 80% of the human genome that is transcribed, only 2% of it is translated into a protein. The remaining 98% of DNA is transcribed into non-coding RNAs such as microRNA, tRNA, and long non-coding RNA. The inability to code for a protein led to the assumption that non-coding DNA is junk. However, recent advancements suggest that non-coding RNA has important regulatory functions such as gene regulation. The non-coding RNA we are concerned with is long non-coding RNA. Long non-coding RNA is broadly defined as an RNA sequence that is equal to or greater than 200 nucleotides long and does not code for a protein. It has similar features to mRNA in that it has a 5' cap, a poly A tail, and undergoes post-transcriptional modification. However, what ultimately differentiates mRNA and long non-coding RNA is the fact that long non-coding RNA does not have an open reading frame, meaning it can't code for a protein. Hence, after transcription, long non-coding RNA folds onto itself to create a secondary structure and interacts with different biological molecules such as DNA, protein, mRNA, and other non-coding RNAs. As such, long non-coding RNA can alter gene expression, protein stability, and regulate biological processes. Recent studies demonstrate a correlation between long non-coding RNA expression and DNA damage repair. DNA damage repair is an essential process as our genome is continuously subject to exogenous and endogenous risk factors that induce DNA damage. An accumulation of DNA damage may result in lesions that impair cell function or cause cancer. Of particular note is radiation exposure. Ionizing radiation is an exogenous risk factor for DNA damage. Hence, we looked at how the expression of long non-coding RNA genes changed in response to ionizing radiation in human embryonic kidney cells, or HEK cells, and we hypothesized that long non-coding RNA expression would vary over time. To perform this experiment, we began with 24 samples of HEK cells and divided them into two groups: sham, which is the control, and a treatment group radiated at 2 grays. A gray measures how many joules of radiation were absorbed per kilogram. We specifically chose 2 grays as it induces 60 to 100 double-stranded DNA breaks to ensure that each chromosome is damaged. After radiation, the groups are further divided according to their collection time post-radiation, 1, 4, 8, and 24 hours. Once RNA was extracted, we performed cDNA synthesis and preparation for real-time quantitative polymerase chain reaction. RT-qPCR is a popular method to determine gene expression. We specifically looked at 4 long non-coding RNA genes. These long non-coding RNA genes were previously studied in HEK cells but were radiated at 8 grays and extracted at 24 hours. However, we were interested in investigating how time influences long non-coding RNA expression. Thus, we investigated how long non-coding RNA expression changed when radiated at 2 grays and extracted at 4 time points. The figures demonstrate long non-coding RNA expression of the radiated samples relative to the sham samples. The sham samples are represented by the dashed line at 0. The asterisk indicates a significant result where the p-value is less than 0.05. As demonstrated by figure 1b, we identified a significant change in long non-coding RNA expression at 24 hours when radiated in HEK cells. These results demonstrate our preliminary trials, and we hope to perform RNA sequencing to investigate other long non-coding RNA genes. From these results, we can conclude that there is a temporal change in some long non-coding RNA genes in response to ionizing radiation at 2 grays in HEK cells. Long non-coding RNA is an epigenetic regulator that responds to environmental stressors and may influence signaling networks related to DNA damage. Hence, long non-coding RNAs may serve as a biomarker for the diagnosis, treatment, and prognosis of cancer and other diseases. In regards to future directions, as mentioned, we hope to perform RNA sequencing to expand beyond the 4 long non-coding RNA genes. Additionally, we hope to expand to different cell lines, including lung fibroblast cells, bronchial epithelial cells, and primary human bone marrow-derived mesenchymal stem cells. Thank you for listening to my presentation, and I would like to thank Dr. Tharmalingam for his guidance and NHRC for providing material and financial support.